Okay, good evening. I would like to welcome you today to our fourth annual evening of science lectures. We prepared some questions for you. I hope you have time to answer them, and we would like to see the polls a little bit later and inform you how many people join us from which corner of the world. My name is Ivica Labuda. I am a science chair for Slovak-American Cultural Center. As I said, I would like to welcome you tonight. And Slovak-American Cultural Center is a non-for-profit organization which was founded in 1967 and preserves Slovak heritage and brings cultural and scientific events to many of us. In the past, we had evenings where we focused on Alzheimer, IT, flu, science and music. We had also discussed science of money. This year, as we are in the middle of pandemic, we had no choice. We had to simply talk about vaccines. What they are, would they really stimulate immune system? And from the standpoint of a clinician, are they safe? Do we trust clinical trials? And I am sure that you will come up with many other questions to ask. So please proceed and write them in the chat box, and we will try to go through all of them and answer them. Before we proceed, I would like to express my tremendous gratitude to all scientists and medical professionals who work tirelessly on developing either cures or vaccines against COVID virus. So I am also very grateful to my esteemed colleagues who are joining me tonight for having, to spend time with us, because they are working nonstop, Saturdays, Sundays, evenings, nights sometimes. So I am really very grateful that they are with us tonight and will give us some light or shed the light on what is in stores regarding vaccine. So first we will start with about 20 minutes presentation by my colleague, Dr. Viliam Pavliak. Then we will bring other fantastic professionals, extraordinary professionals, to the roundtable or panel discussion. So Dr. Viliam Pavliak comes from Gates Foundation. Then we have Dr. Veronika Chromikova from Biocytogen, Danka Pavliakova from Pfizer, and Dr. Martin Malachovsky is an internal medicine doctor practicing in New York City. So let me start with Dr. Viliam Pavliak, who is an expert in vaccine development. He has had 30 years of very productive research done in the area of vaccines. He comes to us from the prestigious Bill and Melinda Gates Foundation, where he is a leader for vaccine development, vaccine process development and manufacturing. Prior to Gates Foundation, he was a head of the vaccine development at the International Vaccine Institute in Seoul in South Korea, and a director of vaccine research at Pfizer. His research before that was done on many also private companies, but also at NIH and NRC. Please, Viliam, lead us into the world of vaccines.
Thank you, Ivica, for a very kind introduction. I am very happy to share with you some knowledge I have about vaccine development, about the COVID. Evening or dobrý večer. I'm really happy to give you some background about different technologies and different vaccines which are being currently developed against the COVID-19. But prior to that, I would like to give you some history about vaccines only on 2 slides. So the first major discovery for vaccine development was done at the end of the 19th century by Louis Pasteur. And actually it happened as a negligence in the lab. It was not him who was who made a mistake, but some colleagues in his lab were preparing to do the experiment and do the inoculation of chicken with avian cholera. The colleagues left the culture on the exposed to the air and light, and then they inoculated the chicken as it was planned. However, the chicken didn't get sick. So they prepared a new fresh culture, immunized or inoculate the chicken with a fresh avian cholera culture, and chicken were protected against the avian cholera. So this was the conclusion for the experiment. After that, Louis Pasteur did a lot of additional experiments to confirm it, was that that weakened microbes can actually provide immunity to infectious disease. So after the end of 19th century, last century was actually a century where a lot of new technologies were discovered, and when you can see those different discoveries on the left, these are different technologies which allowed the development of vaccines. And over the years from the beginning of 1900 up to 2010, there is many vaccines which were developed, and many of those are still being used to save lives, children, adults, elderly people. So let's just give you some idea about vaccine development field. So now let's get to topic of tonight. So this beautiful picture is coronavirus SARS-CoV-2, which was identified as a cause of the outbreak of a respiratory illness first detected in Wuhan in China at the end of 2019. The illness was called as a coronavirus disease, and 2019, and the short is COVID-19. Chinese scientists published sequence of a virus beginning of January, and that actually triggered intense global activity, R&D activity, to develop vaccine against the COVID. So this nice and pretty electron micrograph is showing a three-dimensional picture of the virus, and this pretty red and yellow sticks are actually the surface proteins, which are actually reason why it is called coronavirus, because they show virus as looks as a crown. But also these proteins, especially S protein, is reason why people get infected. So if you look on the infection, so this is the virus, and you have this nice spike proteins on the surface, and those are actually recognized by the ACE2 receptor on the human cells, and that's how the virus is then engulfed into the cell, and then RNA is being released. Proteins are gonna be, the spike proteins are gonna be produced, and a new virus is being formed and released. So that's how the disease actually virus is multiplying inside of body. One of the reasons why young population and children are not infected so much with the virus is because they either lack or they have a very low amount of this ICE2 receptor, while the elderly and older people have more of these receptors, so they are more susceptible to the disease. So now, when human body of the young or healthy person see this virus, it is recognized by immune system, and there are these antigen presenting cells which will then digest the virus and express peptides on the surface, and those then are recognized by T helper cells, which are the cells which then activate the B cell, and the B cell are actually producing the antibody response, which then binds to the virus. Antibody will bind to the virus and neutralize it and kill it. There is another mechanism how immune system works against the viruses, and there is also the T cell immune response. So in addition to antibodies, there are also T cell, cytotoxic T cell activated, and they produce cytokines which then help lyse the virus. So this is the immunity generated by a natural infection. And as we are getting older and older, our immune system is not as good as when we were young, and we are getting more susceptible to the disease. So now that's where the vaccine development comes. So the purpose of the vaccine is to instruct the immune system to produce either antibodies or these T cell cytokines by cytotoxic T cell to kill the virus. The response to the vaccine should, the preference is that the response is stronger than to the natural infection. And also with the vaccine, there are no or very minimal health consequences. So currently, there is around 165 different vaccines being developed for COVID-19 around the world. However, only, as I read it yesterday, 45 are actually in the clinical trials. If you look on the technology or the platforms of these vaccines, so there are eight main platforms which are being currently used for vaccines which are in the development. Those are viral vectors. I will discuss later more. Nucleic acids, which is a new technology. Protein-based, which is protein which is isolated from the virus. And then there are the virus vaccines which are inactivated or could be weakened. So these are the major platforms which are being used for development of these vaccines. So now I'm going to go into more details. So we'll start with the virus. Weakened virus. This is a very similar technology like was used by Louis Pasteur for cholera by accident. Here it is done intentionally. So the virus SARS-CoV-2 is being passed through either animal or human cell lines, that it will lost its infectivity. So it's not infectious anymore. After immunization of the vaccine like this, virus will replicate and induce immune response similar way like a natural infection, but it is not going to cause COVID disease. There is a company in the United States, Codagenics, which is developing Codavax COVID vaccine in collaboration with Serum Institute of India. And this vaccine is actually not injectable, but it's intranasal. Another approach with the whole viruses is inactivation. So the vaccine, the virus is actually grown in the cell culture tanks to the high densities, and then it's inactivated either by chemically, by formaldehyde, or by heat. There is high risk of production of vaccine like this because or companies has to manufacture and grow this virus to the high densities, and if there is some leak, that will cause the disease. There is a company or a collaboration between the Chinese company Sinovac and Butantan Research Institute in Brazil to develop a vaccine like this. The second, another approach is using the pieces of virus, and those are those proteins which I showed you on the electron microscopic picture. Those are those red spike proteins or M protein, which are being used as a vaccine antigens. Since the proteins are being purified, to get a really good immune response, they need some help. So adjuvants are being used to are combined with the vaccine, and they help to induce a high immune response, both antibody and T cell. And Sanofi with GSK is developing vaccine like this. They use the spike protein, those, as I said, those nice red protein, surface protein antigen, and also M protein in combination with a very potent AS03 adjuvant, which is proprietary for GSK. Another approach for this protein-based vaccine is using nanoparticles. That's the technology which actually present the protein on the surface the same way like a virus, but it's not a virus. It's a particle which is not infective. The company in Maryland, Novavax, is developing this vaccine, and also they are including adjuvant, matrix M adjuvant, which is proprietary for Novavax. Third platform which is being used are the viral vectors. Viral vectors are weakened viruses, not COVID or not SARS viruses, but for example, adenovirus is being used broadly for this approach. And into this viral vector, then the genetic information representing a spike protein are introduced. Since this vector is still replicating, so after vaccination, this virus replicates inside of body, so there is more virus produced, and genetic information is then produced to the protein, a spike protein, and immune response is then induced against the protein, both the T cell and B cell. This technology is established, and it's being used for vaccine against Ebola. And there are two companies who are in the later development of this vaccine. One is AstraZeneca with collaboration with University of Oxford in UK. And this vaccine is actually now on hold in US, but is approved. The studies are being done in around in the rest of the world. Another vaccine using this approach is the Russian Sputnik 5. This vaccine is registered and approved in Russia. There is some criticism that there is not enough safety data to approve it, but it's up to Russian regulatory agencies to if they approve it for their citizens or not. Another vector-based vaccine is non-replicating viral vector. So what is different between these two is that this virus, which is again the adenovirus from chimpanzee, is not replicating. So the genetic information is there, and then it's the people are vaccinated with it, and the immune system cells will take up genetic information for this spike protein and produce the protein inside of the body and then which is then picked up by immune system and provide the immune response, both T cell and humoral antibody response. The vaccine, which is being in phase 3 clinical trials, is from J&J, and that this vaccine also is now on the hold. I think last week there was some neurological findings. We don't know if it's actually related to the vaccine, if the subject got vaccine or placebo. However, it's under investigation. So the last technologies, which are actually the most modern ones or latest one, are nucleic acid vaccines. There are two kinds of nucleic acid vaccines. There are DNA vaccine and RNA vaccine. Inovio Pharmaceuticals, which is company located in Philadelphia, they are pioneers in the DNA vaccine, and they have actually a lot of experience in developing vaccine against the MERS in Asia. So how this vaccine is administered by electroporation. So there are guns which actually disturb skin, and that's how the DNA is administered into the cells. Once it enters the cells, then mRNA is produced, and mRNA then is taken by machinery in the human cells, and they use that information to produce this spike protein, which is then again taken by antigen presenting cells and through the B cells for antibodies and T cells for cytokines. The immune response, hopefully protective, is generated. For RNA vaccine, there are two vaccines which are currently being tested in phase 3, and these actually vaccines are the front runners. Pfizer, in collaborations with BioNTech in Germany, they have their vaccine in the clinical trials, and they should have data about the efficacy by end of October, and enough safety data to get the emergency approval by the end of November. Moderna, which is using similar technology, is 1 month behind. So what is different here, this RNA is actually encapsulated in the lipid, and that's how it is, which protects the RNA from digestion by the different enzymes and degradation, and it's used and then it's immunized. Again, same like for this DNA, this mRNA is taken up by cells machinery to produce proteins which represent the surface antigens, the spike proteins on the core SARS-CoV-2 virus, and then immune system will take it up and produce antibodies. So these are the main technologies currently being used for the development. Now, how long does it take to develop the vaccine? It could take from 10 to 15 years. There are different stages, from discovery to preclinical. Preclinical testing is, so prior going to test the vaccine in the humans, it has to be shown that it's not toxic to the animals. So there are toxicology studies done on the animal species, and once vaccine shows that there are no side effects or no toxicology response, then it goes to the phase 1, which is testing in the healthy population, and it's only a few, not a very high number of volunteers. Once it shows safety and immunogenicity in this population, then it's moved to the population which is similar to the population in which a vaccine is going to be used. And then there is more people immunized to generate more safety and immunogenicity data. The final decision about if vaccine works or not is a phase 3 clinical trial, where the more people are being immunized, and the number depends how fast we want to have a vaccine developed, and also on the incidence of the disease. Higher incidence, less people. For COVID, there are 40,000 people being immunized either with placebo, which is usually saline, and then vaccine, and it's blinded. So nobody knows what these people will receive. So to develop a COVID vaccine, it seems that we can have it within 12 to 18 months. However, depending if any of these vaccines will show any efficacy. We don't know that yet, but so far these RNA vaccines are showing a good safety. And this other vaccine from AstraZeneca and J&J, after clearing with these experts, hopefully those will go and finish the clinical testing. So going to the history, the fastest vaccine which was approved, it was approved within 4 years, it was for mumps. But if you compare for HIV, it is 36 years and there is no vaccine developed yet. So it's not pretty that straightforward. So I will finish with this slide. So what actually needs to be done to have a COVID vaccine? So trials have to show that vaccine is safe. Trials have to show that vaccine can provoke immune response, and that the immune response will protect people against the COVID-19 disease. Vaccine can be produced at the large scales, and that has to be developed, which is not really very trivial, and currently is being done at risk. And a lot of this risk is actually is paid for by governments. So the manufacturers are developing production and scaling up the risk before we know the vaccine works or not. Then regulators have to approve it before it can be given. And then it's the logistics and challenge how to immunize the whole world. And the different vaccines have to be stored at different temperatures. These RNA vaccines required -80 centigrades, which is not going to be easy, but manufacturers are working on it to so it is possible to distribute and use it for large population. So that's where I'm going to end, and there will be questions to the panel. I thank you for your.
Thank you, Vilo. This is really fantastic. Thank you. You put a lot of effort into making these beautiful pictures. And at this point, I would like to get to the polls. Where are you coming from? And we have a wide range of people: Maryland, New York, residents of UN, Slovak mission, Minnesota, hello Minnesota, Washington DC, Birmingham, Maryland. Thank you very much for checking in. I'm happy to have you. Let's go to the second question, which was, is SARS, yes, very good. 74% knows the answer. Excellent. I am so happy we have such an educated audience tonight. And the third question was, if the vaccine was available today, would you take it? So 50-50. No, maybe some of them are hesitant. And as you learned from Vilo, from Dr. Pavliak, there are many different ways how to prepare it, and we will see and we will discuss it with our professionals about efficacy and also how they feel. So at this point, thank you very much for participation. And I would like to invite my esteemed colleagues to unmask and to join me at the panel discussion. So I introduce to you Dr. Pavliak. I would like to introduce Danka Pavliakova, who is an expert in the area of vaccine diagnostics. She is a senior principal research scientist at Pfizer. Previously, she also worked at NIH. She published and is an invited speaker at international meetings. Really fantastic resource for vaccine diagnostics. Dr. Veronika Chromikova earned her master's degree in Bratislava, and then she went for PhD to Vienna, to University of Natural Resources and Life Sciences. After her postdoctoral position at Mount Sinai in New York City, she accepted a position in private sector, and she just moved to Biocytogen in Boston. And then lastly, but not at least, is Dr. Martin Malachovsky, who is an internal medicine doctor practicing medicine at Bohdai Medical Center in New York City. He received his medical degree at Charles University, and then he did his internal medicine training at Brown University in Providence. He also holds a master's degree in public health from Columbia University Mailman School of Public Health. Thank you very much, Vilo, and thank you very much to all of you for coming tonight. I would perhaps start with the first question to Vilo, who just spoke his heart out about vaccines. Do you think that the different vaccines, based on the different approaches as you presented it, will have a different efficacy or different adverse reaction?
Okay. Yes, it's possible because presentation is different. Also, the vaccines which include adjuvants could have side effects. So the vaccine which I mentioned, the protein subunit vaccine, which developed by GSK and Sanofi, includes a very potent adjuvant ASO3, which is actually used in the Shingrix vaccine. And I don't know if some of you took Shingrix vaccine, and it has a different effect on side effects in terms of pain in the arm. I had a very mild, but I know the people who had a very strong reaction. So efficacy can be different because different vaccine will induce a different level of antibodies, and also what is important, affinity, avidity, and of the particle specific antibodies, and also cytokines which are going to be induced by each vaccine. So there could be differences in the efficacy.
Okay, perfect. Thank you. Danka, I'm turning to you because I cannot imagine, and maybe you can share with us your first-hand experience on how developing this vaccine differs from the other ones in the past, which you worked on. And also, can you comment on the speed of with which, because I think Vilo mentioned that this Pfizer is based on mRNA. So can you comment on the speed and your experience with it?
So one advantage that was playing for the favor of the vaccine that we are developing was that the mechanism of mRNA vaccine was already being at work, meant for flu. And that was quickly adopted by our German colleagues from BioNTech to use for COVID vaccines. So that's the principle, the mRNA. And for the speed, I've been working in the vaccine business for over 20 years. And the way we worked on this vaccine is completely different. I mean, different in a sense, we didn't go home, we didn't have weekends. We were formulating there's many parts how you put vaccine together. And the scientists who put vaccine together tested it in animals and in mostly in primates initially. And then we started testing in the lowest doses in humans. We had, for safety, as Vilo mentioned, you know, you have phase 1 safety where you start with small number of people. We would immunize first 5 people with the lowest dose. And we would measure the first, number 1 is safety, right? So they were following the safety. And then to look at the immunoresponse. And the immunoresponse, it was so critical to get those numbers right away that we had people being immunized in Chicago, and Pfizer would send corporate jet to get the blood from these people after they were immunized, deliver it to our labs in New York, Pearl River, where we would be testing the immunoresponse. It was completely unprecedented effort. So yes, the speed is like that. And with the current clinical studies, it's very fast. And remember, for the vaccine, you have to have all the components ready and you have to manufacture it. So the company actually went at risk and started manufacturing it earlier before we even know, before it was known that it will work. So yeah, that's different.
That's amazing. I'm really kudos to you and to everybody who is working in that sphere. I mean, it's a tremendous effort. So Martin, to you as a clinician, after you hearing this different approaches to vaccines, are you concerned as a clinician that now people will come to your office? I'm just making this question up. People will come to your office and will ask you, oh, I want a JJ, I want Pfizer, I want this, because they are learning about different approaches. And they said, oh, I don't want adjuvant. Adjuvant, by the way, somebody asked, what is adjuvant? And maybe Vilo can explain a little bit later, but let me ask question. Let me put this question out to Martin.
Sure. Ivica, I think my sense is that the media will play a big role in distribution of the information, and people will want to know what are their options. We'll all have to learn details about the results. There will be a huge public health debate. What's the best strategies? The CDC will get involved. And I think that right now there are many unknowns, and that makes us all anxious, especially seeing the amount of vaccine options, the strategies, and we simply don't know yet about their safety and efficacy. And I think we just have to be patient and wait, and the information will come, and then we'll be making decisions between 1 or 2 or 3 vaccines. Of course, I think we'll have to wait, and some of the vaccines will be not available for children, and some of the vaccines will be more effective in certain populations. And so I think we just have to somehow be patient, because I don't think none of us have the answers.
Okay.
Okay,
Thank
so
you.
I'm going to give the answer for the adjuvant.
Sure, yes, thank you.
So adjuvants help to enhance the antibody response, and the widely used adjuvants so far are aluminum salts, which is aluminum phosphate or aluminum hydroxide. Recently, because they have a big influence on these T-cell immune responses, they are being evaluated, and this ASO3 from GSK is really, GSK will not give any information about it, because it's actually a mixture of several compounds. They contain lipids, and those are purified actually from natural source, from bark from South America. I was working with Novavax, or actually the company in Sweden who supplies the adjuvant M to Novavax, and I saw the production, and it's really a lot of extraction to get those compounds. But in the short, they induce help to induce immune response, and depends on the adjuvant. The response can be modified towards the response which is more appropriate for particular disease.
Okay, perfect. Thank you. Thank you very much. Veronika, could you tell the audience or provide audience with a brief explanation about the antibody treatment, which we saw in recent days that the President Trump was given, or some other people are given? Do you think that that's an avenue to go to? Is it feasible? Is it commercially possible? What do you think?
So to start one by one. So first to the antibody treatment, right? The issue at the beginning is that it takes some time for our bodies to make antibodies after they first encounter antigens or viruses and bacteria. And if the disease is progressing really fast and the antibody is not, the organism is not really catching up and producing antibodies to counter effect, that's where you would go for monoclonal antibody therapy or polyclonal antibody therapy. You would want to supply the organism by antibodies made somewhere else, which can meet this virus and can neutralize it or trigger some kind of other effector functions. And to answer the second part, if it's feasible, I would like to answer with a metaphor which I came up with during the conversations with my father. He's a physicist, very educated, which usually means that he doesn't catch up on biology at all. So when I was talking to him about vaccines and antibodies, I tried to tell him, okay, imagine coronavirus as a swimming pool filled with water, and the summer is coming. So vaccine would be like your swimming instructor you make several appointments with during the course of the spring months, and it's usually cheaper. Therapeutic antibody would be a floaty someone throws at you as you are already drowning in a pool because you didn't learn how to swim before. The problem is these particular floaties tend to be fairly expensive compared to the cost of vaccines. So yes, upscaling is feasible to a certain degree, but the cost will be always more than the cost of the vaccine.
Okay, so that's the comparison of the vaccine versus the therapy? Yeah. Okay.
I would add one more thing. In addition to that, you need to actually administer large amounts because you get it diluted. So your blood volume is quite high, and to achieve the same level, you need a lot of antibody to administer.
So it matters how much.
Yeah.
It matters. So could anybody of you comment on that? We had a question from the audience. Do we know why young people have fewer ACE2 receptors? Could you speculate?
I could toss my wild theory out.
Okay, please do.
It's completely personal and completely wild. So the ACE2 receptors, they have a function in down-regulating the blood pressure, which is not necessarily so important for young people, but might be increasingly important as the people are aging. So because many things in a human organism would be regulated on some sort of self-feeding loop, it would make sense that with increasing age, there would be also increasing expression of ACE2. So that's my personal theory.
Okay.
You know, it makes sense to me.
Okay, perfect. I have a question for Martin, and it is, You have been volunteering your medical doctor services around the world in different countries around the world. When you look at the distribution or projected distribution of vaccines, what do you think? How we will distribute the vaccines?
It's a very good question. I'm not sure if the audience or you guys are familiar with something called cold chain, but I think Vilo mentioned temperature and the importance of transporting vaccines at a certain temperature and storing them at a certain temperature, because all the effort of manufactured vaccines can be very easily destroyed by just having the temperature during transport or storage before it's given to someone neutralized, or the vaccine can become ineffective. And so the cold chain in the distribution of certain vaccines is very important. I am not familiar as to at which temperatures which vaccine will be distributed, but certainly imagine you want to vaccinate somewhere, in the countries which have, which struggle with basic distribution and storage of vaccines. So, I have some experience from rural Africa where, the distribution in the warmer climates and storage is a challenge, but certainly vaccines are being distributed successfully in many places and all over the world, even the more remote areas. And I think it's a matter of, having the infrastructure, execution, money, and willingness of people to take the vaccine, have the trust to the vaccine. And so I think globally to vaccinate 8 billion people in a very short time is, I think,
enormous.
I can't even imagine how it would be possible, but certainly will be depending on each country and on the WHO and all the players in the vaccine world, how to counter it. I would wish to probably mention that children are so far not being tested, and they'll be left behind on having the vaccines available for them for another several years. They are not currently being included in the first trials and studies. And so the children under age, either 18 or 12 will still not be getting vaccines for a while, but of course they are at a lower risk, but maybe a remaining vector of the illness itself.
So do you think, what is the risk to young people? What do you see as a biggest impact on younger generation?
So luckily, I guess this virus is gentler to the children, which I guess scientifically it may be linked to the ACE2 receptor, but the children are more likely to have a lighter illness or no illness at all. They also less likely to spread the illness and are not considered the biggest vector. Distributor of the illness. Their mortality for the children is luckily lower. I think I saw data from 2 days ago where even in the United States, I think 86,000 children were so far confirmed to be infected, and their mortality is 0.25% or less, depending on the state, which is luckily very low for the children. I hope it will remain that way. I have 3 children of my own, and I feel that fortunate that the virus has decided to be relatively gentler to the children.
Yes. Turning back to efficacy, Danka, do you know what is the required efficacy by FDA to get the vaccine approved?
So yeah, well, the requirement is minimum 50%.
Okay. So
to calculate efficacy, I don't know if everybody knows. So the vaccine is right now in clinical study. And I think Vila mentioned that you have part of the people are receiving the actual vaccine, and part of the people in the clinical trial is receiving placebo. And right now we are in the stage where we are testing everybody, doing diagnostic tests, and we are counting the disease, the sick people. Now, company is blind to the results. So we know that there was X amount of sick people, but we don't know if they come from the placebo group or if they come from vaccine group. After a certain number of cases is counted, and I don't know all the statistics, and even if I knew, I don't think I am free to disclose. But after a certain number of cases, independent agency will look at these cases, and then will find out if they are coming from either vaccine group or placebo group. So in case all of the cases are coming from placebo, 0 cases from vaccine group, that would show that vaccine has 100% efficacy. And you know, then it's just calculated. If half of the cases are half, then there is certain math how it's calculated. So we are hoping for the best, and you know, once we get the number of cases, yeah, cross fingers, and we'll see.
Yeah, we have a question here. I don't know who would like to answer it. Does history of having been vaccinated with BCG support the immune response to the virus?
Okay, so maybe I can say something about it, because actually BCG is now being tested as a vaccine against the COVID, and it's people think, especially to use in a population like in the hospitals, and the reason is that TB has this surface cell wall components which could induce this T cell response and not specifically actually kill the virus. So that's the reason, and like Gates Foundation is sponsoring several of these studies, and they are being done in South Africa, just to see if BCG will be good vaccine against the COVID.
Okay, perfect. Thank you. So there was another question, which is, are the vaccines being developed 1x in nature, or will people need to be vaccinated each year? So based on your experience, Veronika, with the flu vaccines, which we need every year, the new vaccine, what is your take on this?
So far, because we didn't have the SARS-CoV-2 around for so many years, but so far it seems to be way more conserved than what the flu viruses usually are doing. So I just saw recently article which was starting with, so far there is still only one SARS-CoV-2 virus identified circulating the whole world. There were no significant mutations except for maybe a single nucleotide mutation, but it didn't seem to have any particular effect on the virus.
Okay.
Yeah, though, so it being said, I'm sorry, the most of the, I don't know, maybe Vilo can correct me, aren't the most of the vaccines being developed with the idea of a shot and then a booster shot? So it would be at least 2 shots for the vaccine?
It depends on the technology. The nasal vaccine, which I mentioned, discussed the first, is being only one. It's a nasal administration. The one which are administered as an injection, those are usually 2. But it will be shown by the trials how many shots you need. But this is planned now. I think it's 2 for most of the vaccine.
Okay, thank you. So there is another question which I think any of you or all of you should try to answer, and it is that wow, 4 years is the fastest vaccine. How can we trust the expedite process, which is most inevitably happening for COVID-19? So in a sense, let's say from February or March, the development starts, yet we are, if Chinese and Russians are able to make this vaccine and approve it in such a short time, are we shooting ourselves in the leg? Okay, we need this really rigorous testing and everything, but we will be left behind other countries. Or do you think that it needs to be really tested rigorously to be safe?
So should I take it? I can take it.
Okay.
You know, it depends on the country and the regulators. So Russia decided to approve it, and I don't think that it's actually being used in the whole population, but it's used for the hospital workers and people like that. Also in China, I think the vaccine is approved for people who are high risk workers in the hospital. I think the safety is important, and you know, we live in the country, and you know, the legal system, people will sue everybody, and then these companies who are making these vaccines, they are then liable to pay for the lawsuits. So, and it's not about the money; it's about to show that actually the large amount of people of immunized, the side effects are minimum, and the vaccine is safe.
I think, yeah, I think if I can only add is that I think trust is as everything is developed over time, and so we are going to have first results of the trials, then we'll start giving certain vaccines to a population, and we'll have more numbers and more reports, and as we grow, we will have more information and more data as to the safety and effectiveness of the vaccines. And it's, you know, we were scared, all of us, to death about COVID in March. 7 months later, we all realized that we are still here, and we look at it in a different way. The same thing will happen with the vaccine. Right now, there's a lot of anxiety, uncertainty, a lot of media talk about, you know, how good or bad these vaccines will be, who will take them, who will not. But again, we will, you know, we will have time to, you know, sit down and ponder and see what we know, what we don't know. And eventually, as the amount of people who will get the vaccine and they will be still around us, or we'll start with a higher risk population and they will survive and they'll be better. I think we'll feel more comfortable as the time progresses.
Also, to add 4 years or 15 years, it depends on the technology. As many of these vaccines, as Danka said, that initially the discovery part was already done for flu. So you just exchange, you put a new sequence, and you develop it for that. For this adenovirus, for these vectors, those were already developed and tested for Ebola, for other vaccine candidates. So what is the only difference is that a piece of the genetic information. So if that spike protein is safe and doesn't induce any, you know, response which could be harmful, I think that it's not really unsafe. I think that this data will show if it's safe or not, the clinical trial. Also, if you have the money, like now the government is putting the money into these clinical trials, like only Pfizer is the only one who is funding everything by itself. So to do such a high numbers of volunteers immunized with the vaccine and make this vaccine at the risk, that makes things faster. If it's done by company by itself, some startup, it takes for a long time to accrue enough people. You have to have a high disease levels, these high incidence, to get enough data to show efficacy and safety.
I have one last question, perhaps for you guys, and it is blood type playing any role for choosing vaccine? Veronika, what do you think?
I wouldn't know. I saw some articles speculating that the blood type might influence the outcome of the infection. I don't know how much. To be fair, I only saw it like this week, and I didn't have time to go into it. So I don't know how much studies was behind that statement, but I don't know how to answer.
I don't think we know at this point, but maybe later.
Maybe later, right? So I would like to really thank you, all of you. I don't want to hold you more than an hour, even though we have many more questions to answer, and it was fascinating. Thank you very much, Vilo, for the world of vaccines, and especially the COVID vaccine, that the wide range of vaccines which are in front of us, who knows what will happen and which one we will choose. So I would like to urge everybody for now to wear a mask. So please mask up as we say goodbye to everybody. I guess this is the only one which can protect us, and have a good evening. And thank you again. Thank you very much.
Good night.
Good night, everyone.
Good night.